Hello students, welcome to Vijetha IAS Academy and myself is Lenin Kaki and I am the faculty of Geography and Science and Technology in Vijetha IAS Academy. Okay, so in this video we are going to discuss one small topic with respect to science and technology, that too from biology or biotechnology topic. Okay, that is three-parent baby, because recently UPSC has been focusing more on this biology and biotechnology topic in UPSC Prelims and also in Mains. Okay, and this whatever the topic that I have been discussing now, UPSC has already asked a question or twice a question from this topic in the prelims. So this topic will be very important for both prelims and mains perspective. Okay, so let us see what is this three-parent baby and what are the techniques that are involved in having this three-parent baby. Okay, so we'll see this one from the basics. Okay, so from the picture, if you see, three parents are involved. Okay, one male will be involved and two females will be involved so that we can have a baby. Okay, means in this one the DNA, okay, remember the DNA of three parents are involved in this one. Okay, DNA of three in this technique. Okay, so we'll see how this DNA of three parents are involved in this one. But before knowing that one, why we are going for these three parents, okay, we have to know why we are going for three parents. Okay, so for that one we have to know, say, DNA in our human cell, whatever the cell that is there, okay, the DNA in human cell will be located in two positions. Okay, one is within the nucleus, DNA will be there, and the DNA will also be there in mitochondria. Okay, but this mitochondria, where it is located? In the cytoplasm of the cell. Okay, if this is the cell, this is which is called as nucleus. Inside this nucleus we will have DNA. Okay, means nothing but we will have chromosomes, and the chromosome portion is there. This is called as the cytoplasm, and in this cytoplasm we have mitochondria, and this mitochondria will have a DNA called as mitochondrial DNA. Okay, mitochondrial DNA. So DNA either will be there in the nucleus or DNA will also be there in the mitochondria. Okay, so if there is any issue with the DNA of the mitochondria, okay, if there is any issue of the DNA of mitochondria, then there will be some sort of diseases that are occurring in the humans, which is called as Leigh syndrome. Okay, which is called as Leigh syndrome, which is nothing but a kind of serious neurodegenerative disease. Okay, neurological disorder is there. Okay, so this neurological disorder is occurring because there is some defect in this mitochondrial DNA. Once this mitochondrial DNA is passed to some children, these children are seen with this disease called as Leigh syndrome, which is having some neurological problems. Okay, so to avoid these neurological problems, okay, or to avoid this Leigh syndrome, we are manipulating these zygotes, or we are manipulating this egg, especially egg we are manipulating, because whatever the mitochondrial DNA that is entering into the children's, that mitochondrial DNA is coming only from mother. Remember this point, children are getting mitochondrial DNA from mother only. Okay, from only from the mother, children's we are getting this DNA. Okay, mitochondrial DNA is coming only from the mother. We will see how this is coming, but because of the defect in the mitochondrial DNA, we are getting some disease or neurological problem which is called as Leigh syndrome. So to solve this problem, okay, to solve this problem, we want to modify this mitochondrial DNA. Either we will remove or we will discard this mitochondrial DNA. For that, different techniques are there, and we will see how these different techniques are working for this one. Is it clear? Okay, so there are three techniques, okay, with respect to this thing, which is called as pronuclear transfer. Second one is called spindle transfer. Third one is called cytoplasm injection. Okay, first one is called pronuclear transfer, spindle transfer, and cytoplasm injection. But remember, nowadays this cytoplasm injection is not functioning, means this technique is not being used these days, and only these two are being used, and this is not used because there are some defects with respect to this technique. We'll discuss what are the defects with respect to that technique. Okay, so but before going to see what are these techniques, we'll see what are the basics of this one. Okay, so first we'll discuss pronuclear transfer. Okay, first we will discuss this one, pronuclear transfer. So I am going to take, see, this is the sperm, okay, this is the head of the sperm, this is the tail of the sperm, right? So if you see mitochondria, okay, and this is having around 23 egg, okay, so egg is also having 23 chromosomes, and this mitochondria of the, once these two are fused, once these egg and sperm are fused, they are going to form which one? Zygote, right? Zygote. But if you see this, in this zygote we are having this 46 chromosomes, because 23 are coming from the sperm and 23 are coming from this ovum or egg, right? But whatever the mitochondria that is there in the tail, okay, if you see this sperm once touches this egg, this tail part will be detached from the head, okay. Once this sperm touches this egg, the tail part will be detached and only head portion will start entering into the egg. So because of that one, as tail is not entering into the egg, whatever the mitochondria that is there in the tail will also not enter into this egg, right? So this, whatever the tail that is there, the tail will be detached and it will not enter into, and only this head part will enter. That's why 46 chromosomes will form, because 23 plus 23, and as this earlier this one is there, right? This mitochondria will come only from the mother, okay. So because of this one, if any defect is there in the mitochondria of the mother, then that defect will be seen in the children's, okay. From this we can say mitochondria enters only from the mother, and if that is having any defect, then it will lead to Leigh syndrome, which is a neuro disorder, okay. So we'll see how these pronuclear transfer will be done. I hope you have understood this one. Only head part will tail, whatever the mitochondria that is there in the tail will not enter into the egg, right? So we'll see. We'll take 2 eggs, we will take, okay. 2 eggs are ovum 1, second one, we'll take 2 eggs or 2 ovums. One is from the mother, this is from the mother, and this is from the donor, okay. So this is donor ovum DO, and this is mother ovum MO, right? So in this donor we are having how many chromosomes? 23 chromosomes are there. How many are there here? 23 are there, okay. And this is the mitochondria from the donor, and this is the, I'll draw with other color, this is the mitochondria of the mother, okay. So this mitochondria of mother is defective, okay. Remember, this is, if this mitochondria of the mother is defective, we want to remove this mitochondria of the mother, okay. So for this purpose, what in this pronuclear transfer, what we will do is, first we will fuse, okay, we will fuse this egg with the sperm, and we will also fuse this egg also with the sperm, okay. This is having 23 chromosomes, this is having 23 chromosomes. Once this mother's ovum is fused with egg, or is fused with the sperm, what we are getting? Okay, so we are having the same, defective, but here see, before these 23 chromosomes of the mother, 23 other, before merging this one, okay, complete merger has not yet taken place, then such structure is called as pronuclei, okay, means complete merger of this nucleus will not occur, okay. Such structure is called as pronuclei. So I will take 46 are there, but these 46 are like this, okay. Okay, total 46 are there, but they have not yet merged completely, okay. The structure which formed before forming into zygote, where both the, whatever these chromosomes are there, those 2 chromosomes, when they are not merged completely from father and mother, the such structure is called as pronuclei, okay. So in this case, this is the after fusing egg with the sperm, and in this case also we will get the same pronuclei, okay. So this pronuclei will also have how many? 46 will be there. Okay, 46 chromosomes. But this mitochondria is defective. Okay, but if this continues means it will form into zygote, right? If zygote is formed, with time the zygote will convert into a child. But this child will have defective, as this is defective, the child will get Leigh syndrome. But we don't want this to happen, right? So to avoid this one, what we will do is, what in this pronuclear transfer is, this is the structure pronuclear, right? So in this structure, what they will do is, they will discard or they will remove the pronuclei of the donor ovum. Okay, they will remove the pronuclei of the donor ovum and they will discard this one. The structure will be looking like this. Okay, and this is perfect mitochondria. This mitochondria of the donor is healthy mitochondria. Okay, so this is having no defect, right? So I have discarded the pronuclei of this donor ovum. I will remove this pronuclei ovum and I will try to transfer this one into this donor's ovum. Okay, so what will happen here? So this, whatever this is there, I will try to transfer this one here. Okay, so I'll try to transfer this one here. So 46, right? So if you see these two, this is from father, right? These 23 chromosomes are coming from the father and these 23 chromosomes are also coming from the mother. Okay, but here whatever the DNA that is there in the mitochondria of the donor, that is also having some DNA, right? So that also having some DNA from the third person. Okay, here this 23 is coming from the father and this 23 is coming from the mother, but we are having the DNA from the third person also, that is the donor ovum is having the mitochondria which is a healthy mitochondria. That healthy mitochondria is also having certain DNA. In this case, if you see, we have discarded this one, right? Because we are not using this one, we are removing this, whatever the pronuclei of the mother is there, we are removing this pronuclei of the mother and transferring that one into this healthy ovum from the donor after discarding the pronuclei of the donor. So now we will continue this one process. So with time it will convert into zygote and after that one it will try to convert again back into baby, because we will transfer whatever this one is there, we will back into the mother. Okay, we will transfer this one into the mother's uterus. Once it is sent back into the mother's uterus, it will try to multiply with time and finally it will form into blastocyst and finally it will form and it will give as a child, right? They will give the birth. Okay, but in this case if you see, 23 is coming from the father, 23 is coming from the mother, is there in the mitochondria that is coming from the third person, that is the donor, right? Hence it is called as, whatever the child that is born from this one is called as 3-parent baby, because 3 persons DNA has been involved in this one. One is from father, one is from the mother and the third one is from the donor's mitochondria. Okay, because we are discarding this healthy, or we are discarding this defective mitochondria of the mother and we are discarding this healthy pronuclei and transferring this whatever the pronuclei of the mother is there into the donor's ovum or egg. Is it clear? And we will continue the process of this one after transferring this entire structure into the mother's uterus. Is it clear? So this is technique is called as pronuclei, because we are transferring the pronuclei, right? We are transferring the pronuclei, means here the fertilization has been done before itself. The fertilization has been done before itself and after fertilization has been done, the transfer of pronuclei has been taken place in this case. Is it clear? So we will see the second case which is called as spindle transfer. Okay, I will take the same case here also. So first we will take (23 chromosomes). Okay, here also I will take the same color. Okay, this is the mitochondria of mother which is defective mitochondria, and this is the healthy mitochondria of the donor. This is healthy. Okay, previous case what I have done, I have taken the sperm and I have fused before itself, right? But in this case what I will do is, discard this. I will try to discard the nucleus of this ovum here itself. I will try to discard the nucleus of this ovum here itself. So because of that one, if you see, okay, So because of that one, okay, this is the donor's ovum without any nucleus, right? And I will remove this nucleus of the mother and I will transfer this nucleus of the mother into the ovum of the donor. So because of that one, what is the structure we are getting here? Okay, this is the donor's ovum, and this donor ovum is having whose nucleus or whose DNA we are getting is, this is the mother's DNA. Okay, mother's DNA, and this is donor's ovum, and in this donor's ovum we are having a healthy mitochondria. Okay, we are having healthy mitochondria in this case. So compared to the previous case we are fusing here itself, but now instead of fusing before itself, I am transferring the healthy nucleus of the mother into the donor's egg. Okay, so after discarding the donor's nucleus, I am transferring the mother's nucleus into this donor's egg, because donor egg is having healthy mitochondria. Once this is done, we are getting this structure, right? I will fuse this structure. Now I will with the sperm. Okay, this is the sperm from the father, which is having 23 chromosomes. Okay, so once this structure has been fused with the sperm, we are getting what is the structure we are getting? This is the egg, and in this one we are having 46 chromosomes. Mother, donors, we are having donors, healthy mitochondria will also be there. So here in this case also, how many persons DNA is there? One is from father, second one is from mother, and the third one is from the donor's mitochondria. Some DNA is there. So in this case also we are having how many people's DNA? 3 people's DNA is there. So once this is done, this structure is once again transferred back into the mother, and the time will take, okay, so that it is transferred into the mother's uterus. Once again it will try to multiply, and finally it will convert into a baby. Okay, so in this case also we are having 3 parents involved in their DNA. So this is also one of the case which is called as spindle transfer. Okay, this method is called as spindle transfer. And let us see the last one, cytoplasm injection. Okay, so in this case also I will take once again the same structures. Okay, same mother's ovum, which is having 23 chromosomes, and in this case also donor's ovum, it is also having 23 chromosomes, but the problem is this one. Okay, so this is defective and this is healthy. Okay, so in this case what we will do is, we will try to once again discard this. We'll try to discard this 23 nucleus or these chromosomes from this donor egg. So this is the structure of discarding this chromosome, right? So from the donor ovum we are removing this nucleus, or we are removing this 23 chromosomes, and this is the structure, right? This entire structure, whatever the structure that is there, this entire structure is fused into this one along with the cytoplasm, or we can say I will remove this cytoplasm. See, this is the so because once this nucleus, or once I have removed this 23 chromosomes, whatever the thing that is there here is nothing but cytoplasm. I will remove this cytoplasm along with mitochondria. Okay, I will remove the cytoplasm along with the mitochondria of the donor. Entire thing will be transferred back into this mother's ovum. Okay, so what is the structure we are getting in this case? See, this is the mother's ovum. Mother's ovum is having 23 chromosomes. With this one, mother's DNA, defective DNA is also there, but as I am transferring entire cytoplasm of the donor along with the mitochondria into mother's ovum, we are having the cytoplasm of the donor along with the mitochondria. This is the healthy mitochondria of the donor will also be transferred into this mother's ovum, right? So now in the mother's ovum in the mother's ovum, if I see, I am having both defective mitochondria and I am having healthy mitochondria also. This structure now will be fused with the sperm. This structure will be fused with the sperm. So because of this one, it will form into zygote having 46 chromosomes. Okay, 46 chromosomes will be there. But the problem in this case is I am having both this defective mitochondria is also there, and also I am having this healthy mitochondria will also be there, right? So because of this one, there is a probability that the Leigh syndrome can reoccur, because this mitochondria we are not removing this defective mitochondria, right? Instead of that one, here we are transferring this healthy mitochondria along with the cytoplasm into the mother's ova, and it is fused with this sperm, right? So because of that one, this defective mitochondria is still there in mother's ovum, and this mother's ovum which is converted into zygote is also defective mitochondria. So because of the presence of this defective mitochondria, that there is a probability that the child might be getting this Leigh syndrome, right? So as there is a probability that the child is getting Leigh syndrome, then what is the purpose of doing all these exercises, right? It's wasteful, right? When there is a chance of a child getting this defect, what is the purpose of doing all this exercise? So because of that one, this entire technique has been discarded. Because of this one, we are not using this cytoplasm injection nowadays. Is it clear? So this cytoplasm injection is not being done nowadays, but the other two, pro-nuclear transfer has been, and spindle transfer, both we are doing on experimental purposes. Is it clear? So these are the three techniques that are used in removing these mitochondrial diseases, right? So this is the small topic which I wanted to discuss about 3-parent baby, where 3 parents are involved. Okay, that is nothing but mother, father, and also the donor. Especially the donor egg is taken, and different cases we will use the donor's egg in different ways, which we have seen in this pro-nuclear transfer technique and also in this spindle transfer technique. Is it clear? I hope you have understood this small video. Thank you, students.